Thank you very much for this wonderful opportunity. I'm really pleased to be speaking with you all today, and thank you for the nice introduction. So the title of my discussion today is why patients with cancer continue to have poor outcomes despite multiple new drug approvals. So I have no financial conflicts of interest to disclose. So let me begin by one of very interesting experiences I have had. I have worked in 4 different countries by now, and whenever I move to a new country, the first thing I do is I get a first aid kit box, because just in case. So as I moved to Canada, I was looking for a first aid kit box in Amazon, and I found this kit box, which was good, with good reviews. But one of the reviews, the top review that I saw was, which said, "Awesome, I can't wait to cut myself." So, you know, the existence of a good first aid kit meant does not mean that you would like to cut yourself just to use that kit box, right? So similarly, in oncology, I find that you know, the existence of a new drug does not necessarily mean everybody should be getting the drug, or the existence of a new drug does not necessarily mean we should ask all patients to be exposed to the drug. I see on social media and on news outlets everywhere that whenever there is a new drug approved, very few people think critically about the drug. Very few people think what the eligibility criteria was, whether it's translatable to the patient in clinic in front of me or not. We just start cheerleading and saying, "Oh, this is going to be a wonderful thing. This is a wonderful new drug. I'm so glad that we have a new drug." But we don't look into the data critically. So actually, if I have to say my conflict of interest, then my conflict of interest is that I really want to, you know, actually build a center for bringing common sense back in oncology instead of just cheerleading and overhyping things. I think the first thing we need to do is we need to have proper patient education and physician education about how to think critically about clinical trials and new drugs. And therefore, I'm so happy today that I have this opportunity to speak with patients and patient advocates to share some of the concerns that I have in the field of oncology. So let me first begin by saying a few words about the concept of surrogate endpoints and accelerated approval. So, you know, cancer drugs, as we know, have many side effects. Some of these side effects can be very serious, and some of them can even be fatal side effects. And we also know that these drugs are super expensive. So the question is, why would a patient be willing to take cancer drugs? And this is not a difficult question. There can be only 2 reasons why a patient would be willing to take a cancer drug despite the side effects, cost, and everything. The patient would want to have an improvement in survival or quality of life. Ideally, we should expect both. The drug should help in prolonging survival and in improving quality of life. Even if it does not do both, it should at least do one of them. So here comes the concept of surrogate endpoints. Surrogate endpoints are endpoints which are not these 2 endpoints. Improvement in survival we measure in terms of overall survival in trials, and quality of life we measure as quality of life in trials. But everything else that we measure in trials, except for these 2 parameters, are surrogate endpoints. And some of the examples I have here in this table, one most commonly used is response rate. The other is progression-free survival. Similarly, there are multiple other surrogate endpoints, but the most commonly used ones in advanced setting are response rate and progression-free survival. I'd like to spend a couple of minutes to highlight some of the important aspects in these definitions of response rate and progression-free survival. Although the name says response, what it actually means is we have a certain criteria. So that criteria is that the tumor size should decrease by at least 30% or more to classify as response. So response rate basically means out of all the patients who received the drug, how many patients had a tumor shrinkage of 30% or more. And progression means out of all the patients who were enrolled in the trial, how many patients had a tumor growth of 20% or more. And progression-free survival, although this term uses the word survival, survival can sometimes be misleading because this is not actually measuring survival, but it's measuring the duration until which the tumor starts to grow beyond 20%. So if you ask why 20%, like why 19% increase in size is not tumor progression, and why 20.5% would classify as tumor progression, there is no evidence for that for this. That's just arbitrary designation. Similarly for response, if there is a decrease in tumor size by 25%, it's not response. It's called stable disease. But if it decreases by 35%, then we call it response. These are just arbitrary metrics, and that's the reason why these may not necessarily reflect patient outcomes. So the question is, why do we use surrogate endpoints in trials then? Why not just measure overall survival and quality of life? One of the reasons people cite for why we use these surrogate endpoints instead of the real endpoint in clinical trials is the time it takes to complete the trial and thereby get the drug faster in the market, which makes absolutely sense. We need to have access to drugs as soon as possible. But how much time are we actually saving by using surrogate endpoints instead of the real overall survival or quality of life as an endpoint? Unfortunately, there are many trials which have not, which don't use quality of life as an endpoint, so we don't have robust data on quality of life. So the question is basically, instead of using overall survival, if we use response rate or progression-free survival or other surrogate endpoints, how much time are we actually saving? So this study looked into that particular question, and they found that if we are using progression-free survival as an endpoint instead of overall survival, then the trial was completed 11 months earlier. And if we use response rate instead of overall survival, then the trial was completed 19 months earlier. So there is some saving in terms of trial completion. If you use response rate instead of OS, then you save 19 months. If you use PFS instead of OS, then you use 11 months. But on the other hand, what it means is it's not, this time saving is not as big as industry wants us to believe. Like, it's not a time saving of 5 years, 10 years. It's a time saving of 11 months, which is not negligible, which is important. But on the other hand, what this also means is it's a trade-off between having access to the drug 11 months earlier and not knowing whether that drug is going to improve quality of life or survival, versus having access to the drugs 11 months later, but knowing that that drug is going to improve survival or quality of life, and therefore being able to make an informed treatment decision about whether or not you would want to get the drug for the given side effects. And the big problem with why we are reluctant towards accepting surrogate endpoints as a trial endpoint, despite this saving of time, is that there have been multiple correlation studies which looked at whether improvement in those surrogate endpoints ultimately led to improvement in survival or not. And the data shows that actually there is not much correlation. So if you see, for example, in this slide, in the metastatic setting, then you'll notice that the correlation for most of them is low. The blue bar indicates very low correlation. And for very minority of them, the correlation is strong enough. So if the correlation is strong, that means improvement in PFS improves survival. if that correlation is strong, then obviously there is no problem with using such endpoint in trials, because you would know that you would be saving time, as well as you would know that improvement in PFS would improve survival as well. But if we are testing the same in these settings where the correlation is so poor, then you would just be improving response or progression, but you would not improve survival. And this can happen for a number of reasons. For example, there could be a drug that delays progression, but it alters the tumor biology so that once the disease progresses, then it starts to behave more aggressively than it would have been if the patient was not exposed to that drug. So there could be a number of reasons why a drug that improves progression, that delays progression, may not delay mortality. And so when I speak with people about these issues, some of them point out a very important question that, okay, delaying progression may not delay mortality, may not delay death, but delaying progression would definitely improve quality of life. So isn't that meaningful? Of course, it is meaningful to improve quality of life, but the question is, as I said, because we have such arbitrary definitions for progression and response, does improving progression-free survival, does it necessarily improve quality of life? So we actually looked into that question, and we published this in 2018. So where we looked at association between progression-free survival and improvement in quality of life. So first, we found something very surprising. That was, we studied 350 phase 3 RCTs, but 50% of those trials, they did not even include quality of life as an endpoint. So, and of those that did include quality of life as an endpoint, 25% did not report pre-specified quality of life information. So that means we are conducting trials, we are enrolling thousands of patients into trials, but we are not bothering to ask how they felt during the trial, whether their quality of life was improved or not. And this was very astonishing to me. Only in these RCTs that I looked into, there were a total of more than 125,000 patients who were enrolled in these studies, and whose quality of life information we don't know. We don't know whether their quality of life was improved, stayed the same, or worsened from these drugs. So this is a huge loss of information. These are patients who sacrifice their time and resources to get enrolled into trial, so thinking it would benefit science, but we don't have any information about their quality of life. But going back to the question, does improving progression-free survival necessarily improve quality of life? We found that there was a very weak correlation. So delaying progression did not necessarily improve quality of life. That was a very important finding. And that does make sense if you think about it, because there should be no difference between 19% growth in tumor size versus 21% growth in tumor size about how worse or how good you feel. But one would be classified as progression, one would not be classified as progression in clinical trials. And the other thing is we need to, I think, distinguish between symptomatic progression and non-symptomatic progression. There are progression that leads to symptoms. If someone is having pain in the abdomen because of metastasis, a big tumor in the liver, then it is meaningful to shrink that tumor. But if there are asymptomatic, there are small nodules in the lung that are not creating any symptoms at all, then probably it doesn't matter in terms of quality of life whether that tumor is 2 centimeter versus 2.2 centimeter. So we talked about surrogate endpoints, but now let's talk about overall survival itself. So if a drug improves overall survival in clinical trials, can we be certain that that is a very good drug? Let me cite an example from Sarcoma. This was in 2016 when a phase 2 trial of Olaratumab was published in Sarcoma, and this was a sensational news at that time because if you can see this graph, the addition of Olaratumab to doxorubicin, it improved overall survival. This is not surrogate, this is overall survival by nearly 1 year, 14 months versus 26 months. So improvement in survival by 1 year, this is something you rarely hear in oncology. Improving survival by 1 year, that was phenomenal. So people said this was unprecedented, which is true. It was unprecedented. We never see 1 year improvement in survival. So this was such a big news that, this led to change in all treatment guidelines, and obviously all the regulatory authorities, most of the regulatory authorities approved this, and even like UK, which is known to conduct robust cost-effectiveness analysis and wait for data to mature before providing funding to new drugs. Even UK allowed early access to this drug because it makes sense, and it's like if someone sees that there is a drug that could improve survival by 12 months, then everybody would want this. But last year at ASCO meeting, we heard the results from a phase 3 trial of the same drug in the same setting, and this was the news that came out. Unprecedented survival benefit vanishes in confirmatory cancer trial. So what we had seen before in a phase 2, this 1 year survival benefit, this was gone. So why does that happen? Because this was a phase 2 trial, and phase 2 trials are limited by various limitations. So what we see in phase 2 trial, what we saw in phase 2 trial was a false positive signal. This drug had absolutely no benefit. In phase 3 trial, there was no benefit at all in overall survival. And this drug, this happened in 2019. So actually, this is a very good example of a good practice. I actually applaud the researchers of this trial because, as you can see, this phase 2 was published in 2016, and this drug received accelerated approval from the FDA, and people had access to these drugs. And just in 3 years, in 2019, they completed the phase 3 trial, and the phase 3 trial showed that there was no benefit at all. And even in these 3 years, we already spent more than $500 million in US alone in this drug. But so this is a good example. This is how accelerated approval should function. First, you give approval on the basis of a surrogate endpoint or preliminary evidence, but you ask that phase 3 be conducted in time, and the phase 3 should be conducted in time. And if the phase 3 says that the benefit was not there, then you withdraw approval. But the problem is, currently, there are many olaratumabs out there that have not been tested in phase 3. Now, you can just imagine, if this same drug was not tested in phase 3 at all, we would be exposing hundreds and hundreds of patients every year to this drug with serious side effects, costing lots of money, and it would not have done any benefit to any patients. It would have done some harm to some patients. And we spent $500 million in just 3 years. In 10 years, you can imagine how much we would have spent. But the problem is there are many other drugs out there that were approved similarly but have never been tested in a phase 3 RCT later. So we don't know whether they would have, whether the benefit from those drugs would have vanished similarly as well. So we talked about, so this is actually not a rare phenomenon, that what you see in phase 2 trial is a false signal, and phase 3, when you do a proper big trial and randomize, then that benefit could actually be lost. So we published this paper very recently where we looked at targeted drugs, because nowadays precision oncology drugs have transformed the world of oncology, and most of us believe that these are really wonderful drugs, so wonderful that they should not be tested in randomized trials and should be approved on the basis of non-randomized trials, because we see very good response rates and very long duration of response with these drugs. But we tried to see if that was actually the case, and we checked the responses in non-randomized trials and compared that with responses of the same drug in the same disease, in the same setting, in a randomized trial. So this is for response rate. It is not significant, but the general outline shows that the responses are higher in non-randomized trials than in randomized trials. But what is significant is the duration of response. If you look at the duration of response, then the duration of response is significantly higher in non-randomized trials than in randomized trials. So that means it's not only about overall survival, but all the parameters like even responses are falsely exaggerated in a non-randomized trial. So if you do a non-randomized trial and you see that there is a duration of response of 14 months, then probably that's a false positive overestimate. If you did the same trial in a randomized fashion for the same drug in the same disease setting, then you would probably see a much shorter duration of response. So this is just a caution about not being misled by seeing highly positive responses or duration of response or even overall survival in a non-randomized or a phase 2 trial. So finally, let's talk about the real gold standard endpoint, overall survival in randomized trial, overall survival in a phase 3 trial. So if a drug shows that it improves overall survival in a phase 3 randomized controlled trial, then is that drug really wonderful? So the answer is it depends on how good the trial was, how good the methodology of the trial was, what were the inclusion criteria. We should be looking after all those parameters before making such conclusions. So I'll give you one example from liver cancer. So this is the first drug that was approved for liver cancer, Sorafenib. This was compared against placebo. And this improved survival by nearly 3 months. So placebo patients had overall survival of 8 months. Sorafenib had nearly 11 months. So this was an improvement in overall survival by 3 months. And this was the first drug to be approved in liver cancer, which is wonderful. But later, researchers studied how good Sorafenib was in the real-world patients. That was trial patients, but what happens if we use it in a day-to-day clinic? So what they found was, first they used 60-day landmark. This is because some patients may be very sick and they may die very soon, so we have to exclude those patients. But once that was done, it was seen that for patients who got Sorafenib, the median survival was 3 months plus the 60 days, so that would be 5 months. And for patients who did not get Sorafenib, it was 2 months plus 60 days, so that would be 4 months. So the surprising thing here is, in the real clinic, patients who got Sorafenib lived for 5 months median. If you look at the trial, patients who got placebo had a median survival of 8 months. So patients who got the drug in real world, they have inferior survival to patients who got placebo in the trial. Why does this happen? Why do patients who get the drug live shorter than patients who get placebo in the trial? Because the trial was highly selected. These are patients with liver cancer. Patients with liver cancer have liver function determinants. Their bilirubin is high and all those things. But this trial had included only those patients with excellent liver function. So these were the best of the best patients in terms of fitness and health. So these are the patients who would live for 8 months even without treatment. But in the real world, most of the patients have a number of comorbidities, a number of complications. So when you use the same drug that was tested in trial to the actual patients with the multiple comorbidities who we see in the clinic, then the outcomes are much, much poor. Actually, I published this paper a couple of months ago in Journal of NCCN, where I looked at all the drugs that have been approved for liver cancer, and this is, I guess, very educational in that we say the median overall survival for patients with advanced liver cancer is 6 months, but here we can see that for the drugs which are approved in second line, so these are patients who have already failed one treatment, even in second line, even in control arm, these patients have a median survival of 7 to 8 months. So this is something that's quite unrealistic in real world. So this shows how selectively patients are enrolled into clinical trials. And that's the reason why even overall survival in randomized trials was considered as surrogate endpoint by these authors and their argument was the overall survival benefit we see in randomized trials does not translate to overall survival benefit for the patients we see day-to-day in clinics. So even when we think about benefits and harms from the drug, we need to think in terms of the overall survival we saw in the randomized trial, also as if it was a surrogate for the real overall survival we would expect in our day-to-day patients. So the question is, we have discussed a lot of limitations of surrogate endpoints until now. So the question is there any role of surrogates in regulatory approval then? So I would say there is, and I think, and I have been on record saying I'm a big fan of the accelerated approval pathway that the FDA has. It is a very good pathway in theory because, you know, it's a compromise between speed of approval and demonstrating that the drugs we have approved are actually useful. You know, even in terms of patients, I can't make a generalized statement, but broadly we have two groups of patients. One group of patients they have a higher threshold for trade-off, meaning by one group of patients tell me that they are happy to accept uncertainty. They are happy to accept side effects if there is some chances of benefit, which is perfectly reasonable. If there is some chances that the drug is going to help me, then I would not want to wait for demonstration of efficacy in a randomized trial. I would like access to the drug as soon as possible. But then there is the other group of patients who say, if this drug is not going to help me, if you are not sure that this drug is going to help me, then I don't want I don't want myself to be exposed to this drug. I don't want the side effects. I don't want to come to cancer clinic every week to get this drug because that would disrupt my quality of life. If you are sure that this drug is going to help me, then I'm happy to do that. But unless you know that this drug is going to help me, then I don't want to get this drug. So those are the two spectrums of patients we have, and accelerated approval is a nice compromise between those two groups of patients, because accelerated approval says, okay, we'll approve your drug on the basis of surrogate endpoint. We don't have to wait for overall survival. We'll approve your drug on the basis of surrogate endpoint. But you have to make a commitment that after the approval, you will conduct a proper randomized trial and you will show that this drug is actually beneficial. If it is not beneficial, then we are going to withdraw the approval. So that's a nice compromise. But the problem is, in practice, what's happening with accelerated approval? We published this paper in JAMA Internal Medicine in May 2019, and in this paper we found this. We found that of the 93 cancer drugs that were approved on the basis of this accelerated approval pathway, only 62%, only 58 of them had confirmed benefit in a randomized trial later. So for nearly 40% of the drugs that were approved on the basis of this pathway, they don't have a confirmation of benefit in a proper randomized trial after approval. But even for these 62%, it's the percentage of drugs that actually improved overall survival in a randomized trial is only 20. What about the other 40%? So this is where it gets a little sad, because the other 20%, they improved surrogate outcome in a post-approval randomized trial. So that means they use, in 20%, they use the same surrogate outcome in the randomized trial post-approval as they had used before getting accelerated approval. So that's not a confirmation of benefit. That means you saw that the drug improves response rate, you got accelerated approval, but in a confirmatory trial, you were supposed to show that this drug also improves survival, but what you did was you again showed that this drug improves response rate. So that doesn't make much sense to me. That's not confirmation of benefit. That's just confirmation in surrogate. And in the other 20%, they again proved that the drug improves surrogate, but in a different surrogate. So got accelerated approval showing that this drug improves response rate, but then in a confirmatory trial demonstrated that it improves progression-free survival. So the percentage of drugs that actually improved overall survival was only 20. And the other sad thing that we found during this study was that for a number of drugs, there were no post-approval confirmatory trials at all. Or some of the trials were, some of the drugs were in the market without confirming clinical benefit for even 10 years. And now we can just think in terms of the same Olaratumab sarcoma example I cited earlier, which was published within 3 years. So that's an excellent example of how accelerated approval should function. Got an approval because the signal was very good, did a confirmatory trial in 3 years, published results, was negative, and the approval was withdrawn. But for many of these accelerated approval drugs, they have been on market for many years without ever being tested in a randomized trial. So for these drugs, for 8 years, for 10 years, we don't know whether they are going to help patients at all. And the other part of this study that surprised us was what happens to the drugs that were tested in a confirmatory trial but failed in a confirmatory trial. By definition, what should happen is if the drug fails in a confirmatory trial, then the approval should be withdrawn. But we found 3 examples of a drug that was approved on the basis of response rate and got accelerated approval and underwent a confirmatory trial. And the primary endpoint for confirmatory trial appropriately was overall survival, but failed in overall survival. So what happens to these drugs? We would think that the approval would be withdrawn, but no. We found that in some cases, despite failing to confirm benefits in confirmatory trials, the accelerated approval was converted to regular approval. The drug went on to receive full approval. So this was very surprising. And for other 2 drugs, the FDA has not decided what to do with the approval yet, even though the results have been published a year ago. This example should be familiar to many of us in the breast cancer community about bevacizumab in first-line metastatic breast cancer. This was one of the high-profile examples where the approval was withdrawn by the FDA because the drug failed to improve overall survival in the confirmatory trial. We recently published this paper where we looked at all the trials of bevacizumab in first-line metastatic breast cancer. We found actually 8 clinical trials of bevacizumab in metastatic breast cancer, and we did a meta-analysis. And we found that the correlation between PFS and OS was inconclusive. So what I'm trying to get here is we have said that if the surrogate has been validated for improving overall survival, then that surrogate may be used. But what this shows is, in case of bevacizumab in breast cancer, we were lucky to find 7-8 clinical trials. Most of the modern drugs are tested in just 1 or 2 clinical trials, and then we don't have enough number of clinical trials to test whether the surrogate has correlation with overall survival or not. In bevacizumab case, we had 7 clinical trials, and even after 7 clinical trials, we were unable to make a conclusion whether PFS was a valid surrogate for OS or not, because as we mentioned in the conclusion, this is a rare case where we have 7 clinical trials, but even that was insufficient to conclude whether PFS was a valid trial level surrogate for OS or not. So what that means is, instead of always thinking about, okay, whether PFS would correlate with OS or not for this drug, whether PFS would correlate with OS or not for that drug, it would be much more expedient and much more efficient to just put overall survival as the trial endpoint. And if we do that, then just 1 clinical trial would be sufficient to say whether that drug improves OS or not. If we are not doing that, then we would not be able to make that conclusion even after 7 clinical trials. So, So what's the solution to this issue of cancer drugs that receive accelerated approval, but they fail to confirm clinical benefit in a future trial? As I mentioned, some of them even went on to receive full approvals. So what can we do about it? FDA is very proud of the number of drugs they approve every year. So they keep posting these pictures, this is from a tweet where it says the FDA approved 2 oncology drugs in 2005. In 2018, there were 48. But we asked this question: is the number of cancer drug approvals a surrogate for regulatory success? So if we are approving more and more cancer drugs, does that actually mean the cancer patients are living longer, are living better? Is that a marker for success? And this is what we found. Astonishingly, we'll see that, of course, this blue bar is the total number of approvals, and that has gone very high in last few years. There is a very nice upward slope. But this purple, which is the number of drugs that demonstrated improvement in overall survival, you will see that these have gone down in last few years. You will see that the number of surrogates, this light blue, number of surrogate approvals based on surrogate endpoints, they have increased quite a lot. So one would expect that surrogates, drugs that benefit surrogates, they receive accelerated approval. So you would think that because the surrogate graph is going up, the graph for accelerated approval will also go up, but it does not go, it is not parallel to surrogate. So what this actually shows is, for example, when the number of drugs, drug approvals based on surrogate is going so high, the number of drugs that are approved on the basis of accelerated approval are in fact going down. So what does that mean? Number of drug approvals based on surrogate is going up, but accelerated approval is going down. That means now FDA is giving more and more full approval based on surrogate endpoints. Now that is a problem, because accelerated approval at least has a filter where they say you should conduct a confirmatory trial in future to confirm the benefit. If you are giving full approval, regular approval, this is the regular approval. Regular approval is going up and up. So that means these drugs do not need to be tested in future at all. They already have full approval. So that's not the proper solution. If we say that these drugs that are approved on the basis of surrogate, they failed in confirmatory trial, what should we do? The answer is not let's give regular approval to all. So at the bottom of all this is the issue of education. We need to, I think, we have failed to educate even physicians and obviously the whole community about these cancer policy issues. We see this lost in translation examples all the time, even in our clinics. And this is actually something that actually happened to me. I told a patient that we have done a brain scan and we can't find anything. The patient said, "What? I didn't have a brain?" Anyways, so there are a number of terminologies that can confuse us, and one of those in regulatory science is that of breakthrough therapies. So, whenever we hear that there is a breakthrough therapy, we assume that it's a wonderful therapy. It's a very useful treatment. If as a patient I had a choice between getting a breakthrough drug versus a non-breakthrough drug, then I would of course want to get a breakthrough drug. Who would want a non-breakthrough drug? But in reality, what breakthrough designated drugs mean is that they are, and what we found in this paper is that these are the drugs that are more likely to be approved on the basis of surrogate endpoints. These drugs have less quality of evidence. These drugs are tested in a smaller trial. So actually, if a cancer drug is called breakthrough, it's more likely to be a lower quality evidence-based drug than a drug that does not have the breakthrough designation, which sounds quite counterintuitive, but In regulatory science, breakthrough means promising. It does not mean established. And similarly, the other is the issue of non-inferiority trial. So a lot of physicians are also confused about what a non-inferiority trial means. So we did this study last year about non-inferiority trials in trials of cancer drugs. So basically, non-inferiority trials, they test whether a new drug is not inferior to the drug that we already have. And how do we define how much inferior is non-inferior? Now, that's a very, very tricky question. The answer is it's arbitrary, and as we saw in this table, different trials use different cutoffs. So 1.16, 1.25, 1.33 even. So what this means is this trial considered, for example, this trial where the non-inferiority criteria is 1.33, what this means is this trial considered up to 33% increase in the risks of mortality as non-inferior. How did they come up with this number? Nobody knows. My point is, did we ask patients how much increase in the risk of mortality is acceptable to them? No, we did not. And without asking patients, how do we know what percent increase in the risk of mortality would be acceptable, would be non-inferior? So one might question, like, why would we even do such a trial where we are not testing whether the new drug is good or not, or better or not. What we are testing is whether the new drug is worse or not. So there are certain scenarios where such trials are justified. For example, if the new trial is testing, let's say, whether we can give this drug once every month instead of once every week, then in that case, patient's quality of life will improve. And if the new drug is not worse than the one we already have, then once a month is better than having once a week. Or if the new drug is cheaper, or if the new drug improves quality of life, or let's say if the new drug is oral, but what we already have is injectable. So in those cases, those are justified. But we found that a lot of the trials, 40% of the trials had no justification. There was no such advantage, but still we were testing in a non-inferiority fashion. We are testing whether or not this new drug is inferior. And if we are using such big margins of 1.33, then we can prove everything is non-inferior. Our trial will always be positive. And you know, like if you put your non-inferiority margin as 1.5, then you'll have almost all drugs as positive. The other thing that needs education to the general public is about all randomized controlled trials are not the same. There are so many ways in which a trial can be biased, and you know, patients can actually sometimes be at a disadvantage by participating in a trial. I'll give you this example from lung cancer. This is a trial that tested first-line Sorafenib versus chemotherapy in ALK-positive lung cancer. So this trial enrolled patients with ALK-positive lung cancer to Sorafenib or chemo. This trial was conducted from 2013 to 2015. But Crizotinib, another oral drug, another ALK inhibitor, had already received accelerated approval in 2011, and it had received full approval in 2013. So this is an oral drug that demonstrated clinical benefit and received accelerated approval in 2011 and full approval in 2013. This new trial was tested from 2013 to 2015. So at this time point, all the patients who would have come to clinics would have received Crizotinib as the standard of care. They were not getting chemotherapy as the standard of care because Crizotinib was already proven to be superior to chemotherapy. But if they got enrolled into this trial, they would get either Sorafenib or chemotherapy. If they were not enrolled in the trial, they would get Crizotinib by default. So this is using an inferior control arm. This control arm is inferior. in 2013 to 2015, nobody should have gotten chemotherapy. If they had ALK-positive lung cancer as a first-line treatment, they should have gotten crizotinib. Another example, this is from melanoma. This trial enrolled patients between 2013 and 2015, and this trial randomized patients to this new drug versus dacarbazine. Now, dacarbazine is a classic old chemo that is not helpful in melanoma. Ipilimumab was approved in 2011 by showing overall survival benefit versus dacarbazine. Nivolumab and pembrolizumab were approved in 2014. This trial ran from 2013 to 2015 and randomized patients to dacarbazine. This is very, very, this is frankly harmful to patients. Patients would have gotten much better treatment if they were not a part of the trial. So, this issue of inferior control arm was studied systematically in this paper that was published last year, in which the researchers looked at the randomized control trials between 2013 and 2018, and then they found that in most of the tumor types, they have some randomized control trials where the control arm was inferior. And if you use the inferior control arm, then you can show that your drug is better than it actually is. Because today's audience is interested in breast cancer, let's focus on this. There was one trial they found that used suboptimal control arm in the randomized trial. So this trial is actually the trial of olaparib in breast cancer patients with BRCA mutation. So, if you look into the methods, you would not explicitly understand that this was a non-inferior control arm, but there is a very subtle way in which they have written it. But you can see this, that patients were randomized either to get olaparib or standard therapy with single agent chemotherapy of physician's choice. But even though they say this is a chemotherapy of physician's choice, it's not any chemotherapy. The physician can just choose between these three chemotherapy: capecitabine, eribulin, or vinorelbine. We know that for patients with BRCA mutation, platinum agents are helpful. Carboplatin has been tested in some studies and has shown evidence of efficacy for patients with BRCA mutation. So here, by mentioning physician's choice between capecitabine, eribulin, or vinorelbine, what this trial is actually doing is this trial is preventing patients in the control arm from receiving carboplatin. Although they don't explicitly say that carboplatin would be excluded, by saying capecitabine, eribulin, or vinorelbine, this means the physician cannot choose carboplatin as one of the chemo for the patients in the standard control arm. So the patients are not getting the ideal chemotherapy in the control arm. And the primary endpoint in this trial was progression-free survival. And the progression-free survival, there was advantage here. But if you look at the overall survival, then there is absolutely no change. There is no benefit in overall survival. It's not significant either. So this is just progression-free survival versus a control arm that did not get the best possible treatment. And what bothered me a lot about this trial, on top of what I already mentioned, was that the industry that made this drug, of course, I'm not against industry and physician collaboration for trials, but what I'm against is that the industry was responsible for analysis and interpretation of the data. Analysis and interpretation of the data. Why should industry be involved in analysis and interpretation of the data? It goes further. The manuscript was written with medical writing support, which was funded by the industry. I understand industry funding for conducting clinical trials, for gathering data, but the interpretation of the data should not be left to the industry. The writing of the manuscript should not be left to the industry. It's like making me the judge of a competition to choose the best article published in 2019. I would choose my own article. So if the industry is allowed to write the manuscript and interpret the data, then there is no question that the industry would say its drug is the best drug in the world. If you want to read in more detail about the other issues with this trial, then I had published this paper in 2017 when this trial was published in eCancer. It is free to access. You can just Google this. But I'd just like to highlight the conclusion from what I read this trial. When I read this trial in detail, I thought that the name of the trial was Olympia trial. Although olaparib seems to have won the gold with Olympia trial, patients probably have not. But it's the patients who deserve the real gold. And we need to stop celebrating a gold-plated bronze as a true gold so that one day our patients can finally get the gold they deserve. So the other issue that I want to highlight today is that of pseudo-innovation. Nowadays, we are so much into innovation and new things that we forget sometimes that our innovation is actually a pseudo-innovation. For example, in liver cancer, there are two drugs. One is called Sorafenib, the other is called Regorafenib. These are two different drugs. But if you look at the structure of these drugs, listen to this, it would be very difficult for you or for anyone to say that these are two different drugs. They look more or less the same. The only difference is this one fluoride atom here. This is the only difference. So they are virtually the same drug, but one is called Sorafenib, the other is called Regorafenib, and now they are tested in such a way, and the guidelines have been formed in such a way that if a patient progresses on Sorafenib, then the second-line treatment is Regorafenib. If they told us that a patient progresses on Sorafenib, then the second line is Sorafenib again, then we would have thought it is crazy. But they say if the patient progresses on Sorafenib, then give Regorafenib, then now it sounds more sensible. But in fact, these two are more or less same agent. So the bottom line of the problem is that clinical trials that are conducted for regulatory approval may not solve patient agenda. So we had written this paper for the ASCO book last year, and it's again free to access, in which we highlight most of the issues with why clinical trials are not patient-centric. But briefly, I think this is the most important part of the chapter that we wrote. What are the key questions a patient should consider before enrolling into a trial? I'm assuming you'll have access to these resources so that you can read this later in detail, or you can find it in that open access book chapter. But there are a number of questions I think will be very helpful for the patients to ask before getting enrolled into trials, so that just to make sure that the trial that they are enrolling into, they are sacrificing their time and resources to get enrolled into, is the trial that's actually helping patient agenda. So what do I mean by clinical trials that don't solve patient agenda? There are a number of examples. I have discussed some of them already. For example, a trial that is designed from the outset to test clinically irrelevant marginal differences. I'll give you an example. This is a trial that was just a trial of a new drug in advanced ovarian cancer. And if you read the conclusions of the trial, it says this new drug, nintedanib, in combination with chemo, is an active first-line treatment that significantly increases PFS for women with advanced ovarian cancer. Now, this sounds okay. There is a new drug that significantly increases PFS. But if you actually look into the data, it is significant, so they are correct. The hazard ratio is 0.84 and p-value is 0.024, so it is significant. But if you look at what's the difference, then it's a difference of 17.2 versus 16.6 months, or 18 days. So this drug improves PFS by only 18 days. But it is significant. That's true, it is significant. But how did this happen? Because this is what we call overpowering a trial. We enroll more than 1,350 patients in this trial. So if you enroll large number of patients, any marginal difference will look significant. Even one-day difference can look significant. Coming back to the examples of clinical trials that don't serve patient agenda, the other is a trial that is conducted without a rationale. For example, there is a new drug that was tested in a phase 2. The phase 2 was negative. The drug failed in a phase 2, but even then, and the trial went to a phase 3. This sounds crazy, but this happens. We did this systematic study last year in which we found that more than 40% of the negative phase 3 trials were actually conducted without a supporting evidence from a phase 2. And 14% of these were conducted despite a negative phase 2. Now, can you imagine this? It failed in phase 2, but it still went on to be tested in a phase 3. Why does this happen? Because if you can find one positive phase 3, then you can recoup all of the cost, and it's financially there is a financial incentive to keep testing in phase 3 even though the phase 2 was negative. The other example of a trial that does not serve patient agenda is a trial that is a trial report that misleads with particular choice of words or misleads with statistics. I'll give you an example. For example, whenever there is a trial publication, when reporting safety, they use such terms to talk about safety. They say the side effects were acceptable, the toxicities were manageable, the treatment was feasible. But what does these words even mean? What do these words even mean? In these trials, we never ask patients, is toxicity acceptable to you? Were side effects acceptable to you? Acceptable to whom? I published this paper in BMJ in 2018, and this paper created some controversy, but I was really upset by seeing trials where patients have died due to toxicity. So side effects cause patients' death. And even those trials said the toxicities were manageable. How was it manageable? If a patient died from side effects, how can they say the toxicity was manageable? And what I found surprisingly was, I did a systematic study and I found that in these trials that use such words to describe toxicities, actually side effects were higher with the drugs than the control arm. The serious adverse events were higher in 84% of such trials. Fatal adverse event or death was higher in 66% of these trials compared to the control arm. And misleading with statistics, I'll give you again an example from breast cancer. Atezolizumab in triple-negative breast cancer. Now again, I don't mean to say that this drug is not useful in breast cancer or anything like that. What I'm saying is how the trialists can mislead, how the statistics can be used to mislead patients or physicians or the whole audience. Because here it is reported that the median overall survival in the total population was 21.3 months with this new immunotherapy and 17.6 months without the immunotherapy. But this difference was not significant. But they mentioned that if we look at only one subgroup of patients who had PD-L1 positive tumors, then the difference now looks very impressive, 25 versus 15 months. It's a 10-month difference, and now this is significant. It looks fine. But if you look at the methods of the paper, the method says that the overall survival would be tested in the total population, intention to treat population, and if it was significant in the total population, then only it would be tested in the PD-L1 positive subgroup. These things are important from a statistical point of view, because because statistically it was designed to control false positive in such a way that first they would be looking at the total population, and if the total population if the results were negative, then they would stop there. If it was positive, then it would be tested for the subgroup. Here, what actually happened was, in the total population, it was negative. So this next part, which looks very impressive, that it was very positive in PD-L1 subgroup, this testing was not supposed to happen according to the protocol. Am I saying that this drug is not beneficial in PD-L1 subgroup? No, it may very well be. The answer is we actually don't know from this trial. we need to conduct a different trial to test this in PD-L1 subgroup alone, because based on the methods of this trial, we can't be certain of this conclusion. So that brings me to towards the end of my talk. So it's very difficult to differentiate hope versus hype, and the media also doesn't help, because the media news can be very misleading at times. I came across this news, I guess 3-4 years ago, that said in BBC, that said remarkable therapy beats terminal breast cancer. What's there not to like about this? It's a remarkable therapy. It beats cancer, and not any cancer, but terminal cancer. So I got excited, and I looked deeper, and I found that this news is based on this paper. And I looked into the paper, and actually, if you know altmetric score, which is a marker of how much attention the paper has received, this had a huge altmetric score. This means this paper was covered in news in many outlets. So what I actually found was this was based on one patient or patient. It's not even patients. It's not even plural. So this whole news about remarkable therapy beating terminal cancer was based on one patient. This is very, very misleading. So that's why it's important to see or dig deep into the news and evaluate what the news is all about. Otherwise, it can be very misleading. I'll give you one more example. This was from prostate cancer. This was a Lancet trial published in Lancet Oncology about this very new drug, padeliporfin vascular targeted photodynamic therapy. Even the name sounds so exotic. And this was covered in news, said it's a leap forward for prostate cancer. It's a game changer for prostate cancer patients. I looked into the trial data, and what I found was actually the control group is active surveillance. So this means we did nothing, no active treatment, just surveillance, just watching the patient. And this is the new drug. And patients who develop metastasis were zero, even if you did nothing. Patients who died from prostate cancer were zero, even if you did nothing. And this new drug also had zero. Like, you can't improve on zero death. You can't have minus death. So how is it a game changer? There is no game to change. There is no people who develop metastasis in this condition. There is no patient who is going to die of this disease. But now you have a new drug for this, and people are calling it a game changer. So this brings me to a little critical discussion about over-treatment and whether we are helping patients or harming them. And this is a little difficult discussion, especially towards the end of life. This is a paper that was published in JAMA Oncology 4-5 years ago, where they looked at chemotherapy used towards the end of life, and they found that even among patients who had good status, this is not among sick patients, even among relatively healthy, fit patients, chemotherapy use in the last 6 months of life, it was associated with worse QOD. QOD here means quality of life near death. So basically, this means there was an editorial published with this article, and it says, cost aside, we feel the last 6 months of life are not best spent in an oncology treatment unit or at home suffering the toxic effects of largely ineffectual therapies for the majority of patients. So I totally agree with this. I mean, what we are practicing nowadays, it seems it's not evidence-based oncology, but it's desperation oncology. Especially with these new immunotherapy drugs, we are so hopeful that sometimes we forget that it may not be in the best interest to, keep fighting at the very end of life when it would actually harm more than benefit. We actually try to evaluate this in patients with urothelial cancer, and what we found was once the immunotherapy was approved here for urothelial cancer, the use of immunotherapy in last 60 days of life, this is not even 6 months, this is last 2 months of life. The percentage of patients who received immunotherapy in the last 2 months of life increased. So it has been increasing. So we are so much optimistic about immunotherapy that we are trying this immunotherapy as a new treatment. This is not continuation. This is initiation of immunotherapy as a new treatment for patients in the last 2 months of their life. So this is very difficult decision, I understand, and of course predicting last 6 months of life is not easy. We are always wrong, but I guess predicting last 2 months or last 1 month of life, we are more accurate than predicting last 6 months of life, and we are actually, I have seen that we have been harming a lot of patients who wanted to die at home. We have brought them to hospital. We have been admitting them, and they are spending their last few days of their life in hospital getting new chemotherapy or new immunotherapy until the very end of their life. And there are a lot of reasons why this happens. I wrote this paper in 2017, and I personally feel very attached to this paper of all the papers I have written. So I try to explore why we continue to prescribe drugs towards the very end of life, and of course there is no easy or one correct answer. There are a lot of reasons why we do this. But one of those reasons is also, I guess, our over-optimism, as well as the fact that we, especially in the West, we want to fight cancer. We use the term of fighting against cancer or battle against cancer. So, not having treatment would look like a loss. But I guess as an oncologist, the most important skill that we need to have as an oncologist is breaking bad news. I published this perspective in Medscape last September, where I have put in my thoughts about the differences in culture in these 4 different countries that I have worked in, and how patients expect differently, and how physicians are supposed to deal with breaking bad news differently. But I guess having those bad news discussion and end-of-life discussion is critical. Please correct me otherwise, because I don't know the patient perspective. I'm only speaking from the physician perspective, but I guess it would be helpful to patients to have this honest conversation with their physicians, and not just ignore this topic. I just want to bring one obituary of one of the patients, not my patient. I just read this obituary in this link that is provided here. But I was very touched by this obituary. You can read the whole obituary here. I just picked one paragraph here. It says, "Team led a good life and had a peaceful death, but the transition was nasty. And for the record, he did not lose his battle with cancer. When he died, the cancer died, so technically it was a tie. He was ready to meet his maker. We are just not sure the maker is ready to meet him." I guess, finally I'd want to highlight this issue of making, advanced care planning and goals of care and all those plannings in place, and having a proper environment where we can discuss about these end-of-life issues would be as important as any other oncology interventions we talk about. So finally, I want to get back to the question that I started this talk with: why cancer patients continue to have poor outcomes despite having multiple drug approvals. And we discussed all those causes over this last 60-70 minutes, because we don't consider value. Some of the terms are misleading. Recent drugs are mostly based on surrogates, and they are never tested for clinical outcomes. And there is a lack of quality of life data. We use non-inferiority design. We are using inappropriate control arms. We are approving based on non-randomized trials, and we are approving me-too drugs. That's all for today. I hope this was helpful to some of you. I'm happy to take questions.